should have an idea that the tumor, it is also as a foreign body to us. So that's why our immune cells, just like the foreign bodies or the bacteria, viruses, they fight them, they are going to fight these tumor cells. Is that clear? Okay, so with this background, so at the end, you will explain the tumor antigen and the immune surveillance to tumor. Surveillance is always they are they are circulating and trying to find any abnormal cell. So the immune surveillance is the immune system. They are always in motion and in circulation just to attack any of the tumor cells. And the mechanism of tumor escape from the immune system. So now, as you know, the immune system is there to fight these infections. But how can these escape and give rise to those tumors? So there are some mechanisms. Before going on, what is the cause of tumor?
Mutations.
How does the mutation occur?
Either methylation or radiation, chemicals.
But okay, so they are chemical, and there are spontaneous also. Without these, during the cell division, there could be some mutations. So even without these chemicals, there can be mutations which can occur during the cell divisions. So spontaneous mutation, and also due to chemical, physical, viruses, and you already know there is a list of inherited, viral, chemical, radiological, and spontaneous. Now my next question is, what are the control for the cell growth? How is the cell multiplication controlled?
By genes, tumor suppressor.
There are genes.
Tumor suppressor genes and proto-oncogenes.
Proto-oncogenes.
And caretaker genes.
Okay, so there are some genes which will cause the cell to divide, and they are known as
Proto-oncogenes.
proto-oncogenes. They are the normal, the cell growth. So they control all the environment, how the cell is going to grow, and what time it is going to divide, and the cytokines are released, and all the other things. When these proto-oncogenes mutate some abnormality, now they become the oncogenes. So the cell cycle is disturbed. So it cannot be controlled. Accelerated, and it will not respond even when you stop the stimulants which produce the change, which produce the multiplication. Even when you stop that, it will not stop. So this is a tumor. So basically the problem is in these two mechanisms. So basically the cell growth always takes place. They are controlled by two group of genes. One is the growth promoting, which promotes the growth, known as proto-oncogenes, and the second is the growth restricting or the suppressor. So this should be in balance. Whenever there is imbalance, tumor will start. So this is the imbalance. And we have, I will not go into so much detail, there are some genes known as these RAS and the ERB and these. So these are the normally controlling the growth, but now they become abnormal. Similarly, these suppress the growth. So there are genes which cause growth, there are genes which control and suppress the growth. Any problem in these genes which control the growth, so there will be proliferation. Any problem in the genes which suppress the tumor, now it's become abnormal, so it cannot suppress. Loss of function. So among them we have the retinoblastoma and the P53, the tumor suppressor gene. So you should know the few examples also. Okay? So there are growth promoting, growth suppressing genes. Now, how do the malignant cell or the neoplastic cell differ from the normal cell?
The neoplastic cell proliferate than function. Its main purpose is proliferation.
So proliferation,
Yes.
it is uncontrolled.
Yes.
The normal, it is controlled. What other thing you said?
Rate of growth is what I said. Its main thing is to proliferate.
Proliferate.
It's not functioning mainly.
The normal cell, they have some
Functions.
function, like they stop somewhere, but here it does not stop. It continues. Then regarding the function, the normal cell has a normal function. This abnormal cell, it will have abnormal function. For example, it's going to produce abnormal proteins now. Okay? Deregulated growth lifespan, as he said, that in the malignant cell, there is no regulation of growth. It is uncontrolled. Clonal in origin.
One clone. And then
Each tumor cell arises from one. So if we have a melanoma, tumor of the mole, so all the cells in that will be formed from one cell, because one cell divides to 2 to 4 and so on.
monoclone.
So monoclonal. So each tumor is monoclonal, one clone. It proliferates. But our cell is so many clones of cells, different cells. Other tissue affinity. Our normal cells, they have some affinity. The epithelium, they have an adhesion molecule, they adhere to each other. So we have a mucous membrane, they have covering and everything. Here, there is some problem. They cannot stick to one another. They spread metastasis. Okay? So these are the properties. What are the changes in the cell? Resistance to control via apoptotic signals. So the normal cells, if something goes wrong, apoptosis, finish. But these, there's no apoptosis. They go on dividing, even they are abnormal. Change the surface phenotype and markers. There is change in the surface phenotype, the surface markers. Before, it has some different receptors, now it has some different, because of the mutated genes. So abnormal proteins. Structure and biochemical changes. Naturally, when there is change in the genes, there will be change in the structure, there will be change in the function. Now before it didn't perform some function, now it is performing that function. Trends of tumor specific antigens. Now, these cells start producing some antigen which are present just on these. These are tumor specific antigens. These are not present in the normal. And these were not present before when it was normal. Now it is producing these. So as you see, how do cancer cells differ from the normal? There are so many changes now taking place in this tumor cell, which is making it something abnormal. And as you know, the immune system is circulating always to find something abnormal, where is some change. And it comes, recognizes, destroys. Okay? So this is the whole lecture. So you should know the mechanism, how the neoplasm starts, what are the changes. Now we'll go directly to the antigen, because this is where the immune system comes and fights. Tumor specific antigens, which are antigens which are present just specific to that tumor. It is not present anywhere else. Tumor associated antigens, these are antigens which are also expressed on the normal cells, and we'll take the examples. So there are some antigens which is present on the other cell and the normal cells. There are some antigens which are now only on these tumor cells. So tumor specific antigens. Tumor associated antigens. So I use the abbreviation TAA, tumor associated antigens. So these antigens expressed at the stages of normal cell differentiation. Tumor associated, they are normal, and during different stages they are expressed. Although the tumor cell expression is either qualitatively or quantitatively different from the normal cell at that particular stage of the maturation. For example, in the blood cells, you have leukemias, lymphocytes, abnormal proliferation of these lymphocytes. As you know, the lymphocytes, when they grow, there is a different antigens which are being expressed during the normal growth, when they mature in the thymus. So they undergo differentiation and they express different antigen on the surface, and then this changes, it goes to the next stage, then it starts producing some other antigen till it becomes mature, and then it expresses some other antigen. So now it may produce the same antigen which was produced when it was very small. Now it is big, it shouldn't produce, but now it starts again producing the antigen which used to be produced when it was during development. So now this is some abnormal. Although this is a normal antigen in
abnormal amount.
Abnormal amount,
yes.
Qualitatively or quantitatively. So the quantity is too much now increased, or the quality is different. You understand?
Like the human chorionic gonadotropin,
Yes.
it's normal in like
Pregnancy.
but in testicular tumor it may be shown in males. And
Okay.
although it's normal, but
It's normal.
in qualitative.
Yeah. So you understand?
expression.
So there are different now tumors. So just don't take that there will be only abnormality in the structure. Not only the structure, but quantitatively the quantity is increased. Okay? So I told you these are mainly for the diagnosis, the alpha fetoprotein, we will come to that, CA and SEG, they are usually for monitoring the tumor. So these are proteins, again, formed by the tumor. These are normal proteins, but in increased quantity now.
Doctor, all these three proteins,
Huh?
these three,
Yeah.
AFP and HC.
Alpha fetoprotein, carcinoembryonic antigen, and human chorionic gonadotropin. Yeah.
These proteins.
Yeah, these are proteins. These are secreted by those tumors. Product of mutated genes. Now there are mutated genes, they are going to produce some things. Oncogenes and mutated tumor suppressor gene, the one which I showed you before, it produces protein that differ from the normal cellular proteins. So now they are different from the normal. And products of mutated oncogenes such as RAS and BCR, mutated tumor suppressor genes, so these now produce abnormal proteins which are on the surface. They are presented. Abnormally expressed but unmutated cellular proteins. So now these are abnormally expressed. Normal cellular proteins that are abnormally expressed in tumor, tyrosinase, it's a protein involved in melanin synthesis. Although normally it is produced, but very small quantity, it cannot lead to tolerance. Now suddenly large amount in melanoma. So now this has become an abnormal protein. Although it's a normal protein, but the quantity is now so much. So remember, it's not only the change in the structure, also the quantity when it becomes large, it becomes abnormal. And the immune cells will recognize these and attack them. Oncogenic viruses, you know that there are some viruses, not all of them, they also cause tumors. For example, Epstein-Barr virus, lymphoma, nasopharyngeal carcinoma, it produces this carcinoma, human papilloma, cervical. So what are these? They cause mutation in the genes, and then these viral proteins, they start neogenic processes. Okay? So I will not go into so much detail, but I just want you to know, because we are just immune response to tumors. So these abnormal proteins. So these abnormal proteins may be from the oncogenic viruses.
We took the details.
You took the details? Okay, oncofetal antigens. Some antigens, they are produced in the fetus, and when the baby is born, no production. But now it starts again producing. For example, the alpha fetoproteins, carcinoembryonic antigen. So these are normally expressed in the second trimester, but in low quantity. But now when they appear in the adult life, carcinomas. So this is what I'm telling, that these are normal proteins. In the fetus they were being produced. At that time it was normal for them. But in the mature it shouldn't produce. Like now in you it shouldn't be produced. Or in very small quantity. But now when they become too large, it means the cancer is producing too much of uncontrolled. It's uncontrolled. Producing no stoppage. Carcinoembryonic antigen, alpha fetoprotein, another one again in the yolk sac, in the liver, produced in the fetus, now in the adult life. So alpha fetoprotein, if I produce from the liver, there is some cancer of the liver. Okay? Beta human chorionic gonadotropin, again in the female it is trophoblastic epithelium, placenta, normal in the female. Detectable in the males or in the non-pregnant. It means that gonadal germ cell tumor, adenocarcinoma of the ovary, pancreas, so these tumors, these are cells which are present at these sites, they start producing abnormal protein which should not be produced now. But this protein was normal in the fetal life. So, artificial glycolipids and glycoprotein antigens. So there are glycolipids and glycoprotein like gangliosides, blood group antigen, mucins, CA125. So these are all proteins or glycoproteins, glycolipids, mucins. They are being abnormally now produced. So larger amounts. Tumor-specific differentiation antigen. This is a classical example. During the differentiation, this is the lymphocyte. So as it matures, here there is some different antigen, then it presents different antigen at this stage. At this stage it becomes different, at this stage it becomes different, and now this is the mature. So there are series of changes. So now there should be only this type of antigen. But if we do the chemistry and we do this flow cytometry and we find the cells, they have this type of antigen, and there are a lot of cells with this type. So it means these abnormal, undifferentiated, immature cells, they are coming in your blood. Again, this is an abnormal. So remember, these proteins, these altered proteins or quantitatively increased proteins, this is something abnormal. Who's going to produce them? Not the normal cell, the tumor cells. Here comes the immune surveillance. Now the basic thing is our immune system wants to find where this abnormality is, try to attack this and get rid of this thing. Now what are the cells do you think which are going to fight the tumor?
Natural killer cells.
Okay. You took this in the...
With you, yes.
Yes.
Natural killer cells.
What else?
Neutrophils.
Do you think neutrophils?
Macrophages.
Macrophages. Specific immune system. Main contributor. We have the macrophages, we have the natural killer cells, but the specific immune system and the innate immune system, both of them coordinate, cooperate to get rid of those abnormal cells, the factory which is producing those abnormal proteins. Okay? So remember which of the cells? The natural killer cells, the macrophages, cytotoxic T lymphocytes, antibodies, cytokines like interferon, tumor necrosis factor. Now, cytotoxic T lymphocytes, how does it kill?
It will bind to major histocompatibility, complex 1, and then it will produce apoptosis.
Excellent. Did you remember how the T cell is activated? Can the T cell be directly activated?
No.
Can it kill directly? No.
Not like the natural killer cell.
Not like the natural killer cell, not like the neutrophils, some other. They just directly recognize and kill. The T cell cannot. T cell needs some help. The help are from the dendritic cell or the macrophages. What they do?
Antigen. Yeah, cytokine.
They can take, or the other best mechanism is, I told you, where is the MSC class 1?
So rest on
On the
cells.
surface
Which cell?
of macrophage.
Abnormal cells.
MSC class 1 is present where?
Normal.
This was the basic, you took this in the case.
One
Plus 1?
AIDS.
Everybody forgot.
Mm.
Very bad. MSC class 1 is present on all the nucleated cells of the body. Normal cells. So what is the significance? Any cell which becomes abnormal, and you remember I told you MSC class 1 presents which type of antigens? Exogenous, endogenous?
Endogenous.
Endogenous. So now any cell of the body, we have different cells, liver, kidney, muscles. So if this cell become tumorous, abnormal protein expressed by MSC class 1
Yes.
on the surface. And which cell will come to kill this?
Cytotoxic.
Cytotoxic.
Cytotoxic. So before it has time to proliferate, it kills it. So you see, MSC1, I told you, is present on all cells of the body. So it is now any cell which becomes malignant, it expresses its own antigen, and the T cell comes, recognizes and finish the cell before it has time to proliferate. So always whenever we have this transformation, these abnormalities in the cells, we have these T cells, they rapidly respond and kill that cell before it has time to divide and to spread.
But doctor, how does the T8, is it by tolerance that the T8 knows that this is an abnormal oncogenic protein and this is a normal protein?
So
by tolerance?
how does the heat recognize the bacteria?
So tolerance knows that this is ours, normal, and if it's not tolerant, so this is oncogenic and destroy.
The tolerance is only in the development when they recognize our own self-antigens,
Yes.
they are tolerated. But now this is now new protein which is produced just right now. We made this T lymphocyte, it is circulating, and we just become abnormal.
So regardless of source, as an
It will-
exogenous
it
or
will-
from our own cells, if it's not the normal, so destroy.
It is going to be killed. It is just like a foreign body.
Mhm.
Did you understand?
Yes, yes.
So, now the natural killer cell, what happens that it kills directly the tumor cell helped by interferon and interleukin 2. So there are some cytokines which act on these. So these are interleukin 2 activated natural killer cells. They directly attack. And they attack the cells which do not express the MSC class 1. So if it doesn't express, the T cells cannot kill them. Natural killer cells they come. So you see, we have like all different cells, not only the specific, the non-specific. They are also helping in killing that because this is something very serious. We need to kill it. Macrophages, how do they kill? They also directly attack and they kill by production of reactive oxygen intermediates, you know, the superoxide and all those.
Reactive oxygen species, yes.
And the tumor necrosis factor, they produce some cytokines, they are going to directly kill these tumor cells. Cytotoxic T cells, as I told you, so MSE class 1 and the cytotoxic T cell. Now how does it kill? By bullets or by sword, what?
Cytokines that draws apoptosis.
So, by the TNF, by perforin and granzyme, this is the T cell, this is the target abnormal cell, it produces perforin and granzymes, they act on this, kill this. Or by tumor necrosis factor or by FAS and FICE ligands. This produces the ligand, FAS, this is the receptor, it binds the FAS, death signal. So how does it kill the cytotoxic? By these three processes. And you already know that T cell is going to kill only when there is MSC class 1. If there is no MSC class 1, natural killer will help. The macrophages can come, other can come. But the T cytotoxic
Only if there is
needs the
MSC.
MSC class 1. And this is the significance that God has given us MSC class 1 on all the cells of the body. So whenever something goes wrong, this is just a cytotoxic, the tumor cell, the class 1, T cell receptor, FAS ligand, perforin, granzyme. Just to B cells. Do the B cells also help?
Antibodies.
Antibodies. Antibodies, they attack the proteins. This is producing abnormal proteins. So why not? The B cell become converted to which cell?
Plasma cells.
Plasma cell. They start producing antibodies against these proteins. How will it kill?
Destruction.
How?
Opsonization.
Excellent. What else? What is opsonization?
Make the cell marker for other cells to come and destroy it.
This is a tumor cell. It is attached by antibody.
Yes, sir.
I write so much, that's why I finish your markers.
No problem.
How will it kill now? This acts as an opsonin. I told you to make it more tastier. Now, what happens? The natural killer cells, they can come. The macrophages, they can come. These cell comes and recognize these receptor, and now they can kill this cell as an opsonin to make it more tastier.
Like put an X, a marker,
Yeah.
so that can be.
So it can kill by antibody-dependent cell-mediated cytotoxicity. Remember, I told you ADCC.
antibody.
Antibody-dependent.
Cytotoxic.
This is antibody-dependent. Cell-mediated cytotoxicity.
Causes sent to that.
If this was not present, how will it kill? So this is another. So actually, the B cell, they start producing those antibodies, and these antibodies, they bind on those proteins, abnormal proteins. So they can kill the natural killer macrophages. They can kill the macrophages. These can kill even the complement. You remember the complement? How is it activated? The complement. How? What does it require?
But
What does it require? The classical pathway. It requires the antibody. So again, the antibody is helping the complement system. The antibody is not killing. The antibody attached to this, the complement activated, and it produces
the attack complex.
membrane attack complex.
C5.
So you see, we have the innate. Complement is what? Is it specific? Non-specific. Remember the innate immune system, it is non-specific. So the innate immune system is helping the specific immune system to kill. So just see how many mechanisms there are. Innate, the specific, the complement, the antibodies, the macrophages, the natural killer, all of them fighting this one abnormal cell. This is just to show you that what is opsonization. So this is a tumor, it becomes coated with this antibody, opsonization, the macrophages come, or these natural killer cells, the ADCC, the antibody dependent cell. So I just gave these pictures for the text.
Escape.
Now, still we have tumors and we have cancers in the population. What then happens? The immune system is fighting,
Escape.
but ultimately it overpowers the immune system and destroys our immune system. And now the immune system is helpless. All are fighting, they are still fighting. They cannot.
Rate of growth and differentiation exceeds the
Yeah.
rate of
So
fighting.
their army is more powerful
Mhm.
as compared to our army.
Like the bacteria, specific types of bacteria and
Yeah.
viruses. Yes.
We have sometimes sepsis. The bacteria, they become so much, they divide and grow. Our immune system cannot do it. Reduced level or absence of MSC class 1. What will happen? No MSC. The tumor is growing
Yes.
without expressing the antigen.
Yeah.
How the T cell will kill.
No, it will not kill.
So this is escaping. So some of the tumor, they do not produce MSC class 1. So no antigen expression. And the T cell will come and say, oh, it is normal, no problem. It goes away. Some tumors stop expressing the antigen. Antigen loss variant. As you know, it's abnormal protein, it should produce. Now the tumor, somehow, it doesn't produce those antigens
Hidden.
on the surface. Hidden. The T cell comes, cannot recognize. Production of immunosuppressive factors by the tumor. The tumor produces some immune suppressive substances, cytokines, transforming growth factor. The tumor starts producing this, the immune system is suppressed. It suppresses the immune system. Tumor antigen may induce specific immunological tolerance. It produces some more antigen. So you remember that when the lymphocyte is developing in the thymus, these antigen will be expressed in the thymus, and any T cell against this antigen will be deleted. So now they can grow freely.
So like the body will, or the immune system will consider the oncogenic cells as a normal cells
Yes.
and will not attack
They
them.
will not attack. Tumor cells have an inherent defect in antigen processing and presentation. This tumor cell, I told you, it produces abnormal antigen. It has to be bound with MSC class 1, presented on the surface. But now it cannot process, it cannot bind this antigen with the MSC, it is not presented. So it protects itself from the attack. blocking of receptor on the T cell by specific antigen antibody complex. Now, this is the T cell. It has a receptor. This tumor cell, it produces antigen, it goes and binds this. So how can it bind this then? You understood? It throws the antigen and these complexes, they bind the tumor, this cytotoxic T cell. So now the receptors are full. How can they engage this receptor?
So
the
like
tumor
they
cell
send something to occupy the receptors so that they don't bind to
my receptors.
Yes.
So now the T cell, they are helpless. They are just, all the receptors are full. How can it attach to me, which I am presenting?
Like an inhibitor.
Like an inhibitor.
Antagonist, yes.
Ibrahim, are you understanding?
No. Number 6 is enough.
Tell him.
Like the mechanism of antagonist. In pharmacology, we talk antagonist will bind to the receptor and prevent its activation. Here the same, but the antagonist secreted from this tumor cell will bind to the CD8 receptor and stop its activation.
Tell him in Arabic.
I
You
got-
understood?
I got, yeah.
So the T cell, it
Block-
has receptor.
block the receptor.
It has to recognize mine,
Mm.
come to mine. But if I throw antigens, and the antigens, they block these receptors, will not bind.
Yeah, صح.
Blocking. Antigens on the cells of the surface tumors may be masked by sialic acid containing mucopolysaccharides. So if this is the abnormal cell, abnormal protein, it produces
Coat.
coat. So even if the T cell come, hidden,
Mm.
it goes away.
Cures happens with malignant or benign?
Any tumor, any tumor. Benign is controlled. When we stop the stimulus, it will control. The malignant, uncontrollable. Even when you stop the signal, the chemical substance which produce this change, now you stop even this chemical substance, but it will proliferate. But if like something growth factor in the benign tumor, it is causing it growth, you stop the growth factor. These are uncontrolled, malignant.
Thank you very much.
So, did you understand?
Yes.
So it's
Isn't one of the escape is to have the tumor itself in the system? So like if Epstein-Barr virus, we have B cell lymphoma. Isn't that we're affecting one of the immune systems that it will not attack itself? So this is one of the mechanism of escape. What I'm saying, if the tumor was in the immune system, like
the lymphocytes.
yeah, this is a way of escape.
This is already
because it will not attack itself.
the lymphocytes, which are our normal good cells, which are the army, which is helping fighting. When it becomes neoplastic, it loses its function also. So now there are in what happens in leukemia, you have thousands of lymphocytes. Normally you have very less. Now there are even more than that. They don't function. Can I have the attendance sheet and my pen? My pen is Mia's methane real.